Oh, now I'm nervous. I'm not sure if I'm recorded before. Hi, I'm Jody Bourgeois, retired professor from Earth and Space Sciences. I know a bunch of you, some of you from the New Mexico field trip last year. Yes. So it's my pleasure to introduce Professor Dr. Kathleen Campbell. Thank you. Who, as I say, this is partly from what's online, so you can read about it, but she started her career as a straight up geologist, I guess, and went to UC Santa Cruz as an undergrad, and then came to the University of Washington to work with Jody. And I said, hey, Kathy, you're interested in fossils and paleoecology. There's this formation on the coast of Washington to work on. And so that was her master's project, which it's still ongoing. We're still working on papers, but she discovered extremophiles. So this was in the mid 80s, right? It's kind of a new topic, but there were these cold seeps preserved in the Pliocene on the coast of Washington. And so that from there, she's branched out into all kinds of extremophiles, and she'll be telling you more about that. After she went to USC for her doctorate and then to a postdoc at NASA Ames, worked with the late Jack Farmer, whom some of you might have known. And then went to join the faculty at University of Auckland, kind of far away.
Yes.
But at University of Auckland, I think she said she initiated the Center Paleoenvironmental.
Exactly.
All right, the Center for Environmental in school, and she was telling me she initiated an undergrad astrobiology program with a class that spans many disciplines. It sounds kind of like what Jody Deming had done, something like that, like where all the way
Roger
from philosophy, but to art, to
That's right.
everything about it. So she has lots of honors. I won't go through the many honors. You can Google her online. But I'm interested to learn more about her involvement with the Mars project. I knew about the hot springs and the cold springs and the cold seeps, but so this will be new for me too. So Kathy, take it away.
Thank you, Jody. And thank you everyone for coming. I so appreciate it. I'm on a research sabbatical this semester, so I'm away. Someone else is teaching my astrobiology course. We team teach, so there's 7 of us who actually teach it. So someone else is wrangling all the faculty while I'm out learning from all of you and doing research. So I might just sort of jump straight in. And I know that says this meeting is being recorded, but I wonder if we can maybe put that little okay thing off. I don't know if I can touch this. Oh, that says leave meeting. Don't touch that. It just happens to be over the words, that's all. So if that can go, I don't want to touch leave meeting. Oh, Megan, total star. So we're going to, let's launch straight into this. So I do paleoenvironments and paleoecology, and I want to talk about how early life and Megan, I'll just leave it. I know that. Thank you. I know there's people coming in online. So I want to talk a little bit about how my work links to early life on Earth and the possibility for life on Mars. And we will talk about Mars exploration and Mars sample return. So let's see if I can just push one of these buttons. Which one makes this forward? Ah, cool. That wasn't what I was expecting. So there's a large research team, and I'm just listing a few people whose work is highlighted in this talk. And we are international from the University of Auckland, Argentina, all around to Australia and other places, Japan, not shown here, but you'll see later. And so this is a team that is doing fantastic work, and there's no way that I could have done all this work by myself. But I guess I'd like to say that I've contributed as a leader of this group. So let's see what kinds of, whoa, that's scary. I'm just trying to see, oh, this is it. I'm just learning this computer. So what I want to do is I kind of want to break our conversation into 3 parts. And I'm aware that some people are going to be dashing off to a faculty meeting at the end. So that very last one, you might catch part of, but maybe not the whole thing. And I want to get into just super briefly, I don't think this group needs a lot of introductions in terms of life in the solar system, but I will make a few statements that will lead us towards my own research on hot springs, which is what I'm focusing on today as analog environments for the search for both early life on Earth and possibly extraterrestrial life. And then I kind of want to get into this problem of biosignatures, the types and controls on the quality of their preservation, and kind of the debate that goes on in our area around biosignatures. What are they, and how can you find one that you can rely on? So we work on Earth analog environments to help provide the context and narrow the search for extraterrestrial life, past and present, as well as understanding how life on Earth may have evolved. So we are today really going to be focusing on these Earth analog extreme environments. And, you know, life has these three basic requirements: to have the organic building blocks, the energy source to assemble life, which can be a solar or a celestial body source of energy, and then this liquid medium, which of course with NASA, it's follow the water for all this exploration of our own solar system and even out beyond our solar system. So as we know it, of course, we're not talking about life as we don't know it. So the first thing I thought I would do is show you an extremophile from New Zealand. This is the world's, I think, only alpine parrot, snow parrot, the Kia. If you ever come to New Zealand to see the Kia, you'll find Kia in the mountains, descended from the forest kakas. And the Kia is very cheeky. And if you leave your car up there in the mountains, it will shred all the rubber off of it. They're very smart, and they are extremophiles on one sense if we look at the Earth through this idea of the biospace, where we look at these limitations on life, its tolerances to extremes that might be physical or chemical. So we'll see a few New Zealand culprits as we go along today. So I really like this concept. I learned this from Charles Cockle over in the UK, an astrobiologist over there, about life all being inside this big zoo, all life on Earth. And the physical and chemical limits are the fence, and they separate life from death. So there's a few limits shown there, and you can define them in different ways: survival, reproduction, etc. And I think the thing to remember is that then we can look at Earth's conditions and say, is there overlap with these extraterrestrial conditions on other celestial bodies with those that are suitable for life? Because we start with life on Earth as our starting point. And as you can see with the arrows that are going out, that biospace can expand as we discover more extreme environments, certainly here on Earth. And a great example of that I think all of you will be aware of is that you can extend that biospace in all these different directions, and the edge of the biospace is pretty much dominated by microbes. They're well adapted to modify and go through ways and physiological changes to adapt to their environments, and that's harder for multicellular organisms to make those kinds of changes in general, unless you're a tardigrade or a water bear, but otherwise, which are the superstars and the eukaryotes for extreme conditions. And so we're talking about the edge of the biospace being dominated by extremophiles, and they're living in extreme environments like the hydrothermal vent organisms living at 122 degrees Celsius. So that's what we're talking about. And of course, in the 1970s, the edge of the biospace was expanded with the discovery of life at the hydrothermal vents in the deep sea. The geologists went out there on their cruise to the Galapagos. They found all this life. No one was expecting it. They were looking for hot springs. They found the hot springs. There was no biologists on that cruise. So they had to run back, grab some biologists, go back, collect these animals, and find out that they were using Earth's geofluids as fuel for life. And in particular, for example, sulfides coming out of the vent fluids, so geofluids utilizing, you can see the microbial mats on the seafloor on the right, those yellow things, utilizing the sulfides, but also within the tissues of the organisms, being chemosymbiotic and having endosymbionts, then being able to grow without a gut in total darkness, living off these geofluids. So that changed everything. And when the Viking missions went, remember, we didn't even know about hydrothermal vents harboring an entire ecosystem. And there was the Viking missions trying to find life on Mars. So one other point I want to make about hydrothermal environments, because then I'm going to focus in on hot springs on land, is that if we look at the tree of life, we see in the three domains that purple branches at the bottom of the tree of life. So the most primitive living things are heat loving or hyperthermophilic or thermophilic. They love hot temperature. And so this idea that the last universal common ancestor, or LUCA, is that also, was that also hydrothermophilic as well? And if we think, yes, okay, Earth was hot 4.4 billion years ago, we were going through the bombardment phase of the cleanup of the formation of the solar system. So is that a remnant today? Or is it simply horizontal gene transfer between, say, the archaea and the bacteria? And, you know, that's a debated question whether LUCA itself, for example, was heat loving. But hydrothermal vents, hydrothermal extreme environments have been implicated as the place where life may have originated. Now, how many people in this room have been to Yellowstone National Park and seen some of the hot springs? So this is Grand Prismatic Spring, and I'd like to know if any of you know what is wrong with this photo, which I pulled off the internet. It's fake news. What is wrong with this photo? Have you been to Grand Prismatic? Well, maybe you don't know, but boy, at NASA, they sure did when I gave this talk a little while ago. There's no boardwalk here. They erased it. God, you can walk right up to the edge of the spring. It's crazy. So be careful what you find on the internet, right? All right, well, the reason I put this picture here is it's gorgeous. And we see that in 1969, Kastenholz, a microbiologist working at Yellowstone, discovered extremophiles in these hot springs, land-based hot springs at Yellowstone. And all those colors that you see, of course, are different kinds of microbial mats living in these different temperature regimes in these terrestrial hot springs. What I also want to point out to you as a geologist is all the white stuff. The white stuff. The white stuff are actual deposits from the hot springs made out of silica, hydrothermal silica. And it is that material that comes out of the dissolved in hot springs waters, and it starts to cool and it dumps out. And what does it do but coat all of these microorganisms and preserve some of them, trapping them in the minerals, and then leaving us an archive that we can study in the geological record, which is where I come in, and not just me, but others as well. So I want you to notice the sediment, not just the microbes. They're together give us our record that is so important to try to understand why these hot springs are so relevant in the search for life beyond Earth. So here's another picture of New Zealand. I am not being paid by the tourism board, but I got to tell you, it is a very, very beautiful place. And if you haven't been there, come in the summer, which is your winter. You might as well get out of here. I know what it's like in the winter here. I used to live here. And come on over, and this is Orake Karaka, which is one of my favorite places. And you'll see on the right is actually a baby rock on the right because it's only 60 years old, and you see a microbial mat there in that sort of orange color on the right. And that's the UV pigments, the carotenoid pigments that are actually protecting these microbes from sunburn. So the two things about the silica and the hot springs I want to focus on here is preservation and predictive quality, okay? So here we see these living mats. You see all those orange and green and yellow colors, and you see a hot spring there in the summertime almost being overgrown by the mats. And those mats are already rock. They're already infused with silica. If you take them back to my office and you dry them out for a month, they turn pure white. They're made out of pure silica. But the organisms are alive and they're growing and they're trying to outgrow the silica, but they are full of this opal, this non-crystalline material, which is known as sinter. It makes a rock deposit known as sinter. And you can see in that fault-bounded terrace on the left that's white, wherever there is no water, the mats dry out and you get the sinter forming. So it's our geological archive. The other thing about this is that the springs themselves are highly predictive in terms of what kind of morphology, what kind of shapes of these mats that you have. They have very distinctive textures, a type of biosignature, a type of indicator of life that tell us particular things about the hot spring environments. Are you at the vent? Is it boiling? Is it surging? Is there a little thermal spring outflow? Is there a deep pool? Is there a shallow pool? Are you out on the marsh? All the microbial fabrics or textures tell you where you are in this system, particularly in sort of a temperature gradient. And so we can use that to help narrow the search or predict where we might find signs of life, both early Earth as well as perhaps Mars. So I'm going to show you this model, not because we want to memorize it or look at all the words, but I'm going to show it to you because it's a cross section that I've been working on for a long time. Jack Farmer worked on it before this. And for those of you who know Australians, Malcolm Walter also worked on this model of hot spring center forming in association with these mats. And you see on the left, you're near the vent. We're sitting at about 100 degrees, and that's known as the proximal apron. And then as you get away from the vent, you get into the slope, the middle apron, 65 to about 40 degrees out to the distal apron, and even the geothermally influenced marsh areas. And so it's quite predictable what kinds of mats you'll see, and they turn to stone. So the one that I showed you earlier is known as Palisade fabric. And I put a little green tick mark over here where you find Palisade layers on this distal apron terrace. So you know how, in a relative sense, how far away are you from the vent? And so we'll get back to this gradient as we go along and how we sort of map this out and what we do with it. And just to show you how stunningly beautiful it really is, and I can't go into all of it, and it won't be of interest to every one of you, but all these different shapes and colors, and particularly the textures themselves, are stunningly informative, with bubble mats on the left with all those bubbles. That's all from photosynthesizing bacteria, giving off oxygen, and that's being trapped in the mats. It becomes silicified and turns to stone. Just as an example on the left, and I just found a big block of this in Nevada just 3 weeks ago, Miocene in age, and I could not throw that rock away. It was too stunning. I sent it back to New Zealand, which is crazy, but I did, because I have a big collection where I teach students about all of this. The two in the left, this one that's been cut out looks like caramel slice. For the 2 Australians in this room, you'll know what I'm talking about. It looks like a dessert in New Zealand. That's a whole nother kind of mat that grows in shallow water. Streamer fabric grows where there's flowing water, and then we have domal stromatolites growing in the pools. So anyway, there's a whole story around this that we've been developing over a long time. The point is it's predictable, and you can actually take this back into the fossil record. The other thing we do is map. So here you see a spring, Whangapaoa Spring in New Zealand, and you see the outflow channels. There's 2 of them. And we map out their distributions of temperature, pH, and those textures by color. And it's very highly predictable. For example, I'll show you 2 things about this that's relevant for the next things I want to tell you about. Number 1, the big outflow channel that we mapped first has a certain distribution of these textures. And you could predict that the other outflow channel would have the same distribution. And when we went over there, indeed it did. The pink goes to the green, which goes to the orange, which goes to the brown. I won't go into the details. It's very exciting if you're a paleoecologist. So we can predict what we're going to find in those outflows based on temperature and also the conditions. How deep is the pool? How fast is the water? The other thing I'll point out here is this pool temperature at 65.8 degrees, which immediately tells me there's no way we're going to find the proximal vent textures at all, because it's too cold. This spring is not warm enough. And what aren't we going to find? We're not going to find the geysers. There's no geysers here. It's not boiling. We're not going to find the rock type, center rock type called geyserite. So let me show you something about geyserite, because that turns out to be super important for the next story. This spring definitely, predictably, does not have any geyserite near the vent because it's too cold. Here's some geyserite living today. Meaning the sense that this is an active hot spring at Iraqi Caracal, and you can see there are columns, there are little spicules, little small things, there's knobby bits in the back. There's actually 4 different textural types that tell you all about where are you in relationship to the boiling spring at 90 degrees in New Zealand. And so this geyserite, right through the 1970s into the 1980s, was assumed to be devoid of life. It's only gray, you can only see the silica. You can't see any mats. And it turns out, actually, there are organisms living at 90 degrees, at 100 degrees in terrestrial hot springs, but they are biofilm. They're not big, thick cyanobacteria because they don't grow above 70 or even 65. And so we were doing some work in Jurassic centers down in Argentina, shown here. Let me see if I can, oh, there's the arrow. So this is a little spicule of a center. And when you do Raman work on it, you see it alternates. We've got it colorized for carbon, which is the green, and the yellow is quartz. And so you see alternating laminations or thin layers of microbial biofilm and silica. There's life everywhere in hot springs, even at the highest temperature. Okay, so you're like, well, that's exciting. Why does that matter? Well, it matters for a lot of reasons. And one of the reasons it matters is if you decide you want to go look for the earliest life on Earth or among the earliest evidence for life on Earth, it gets very tricky very quickly. And so this is a sample, a rock from the Pilbara Craton in Western Australia. It's 3.48 billion years old. This is a PhD work of Tara Jokic, a student over at UNSW, who's now at the Sydney Museum, Australian Museum, I believe. And anyway, she was doing this field work, and I'll show you a picture of Tara. Okay, move. Uh-oh, move forward. There we go. Oh, we went too far. Oh, now we're going the wrong way. I might need some rescue here. Oh, we're definitely going the wrong way. How do I go backwards on this machine? Nope, we're still going forward. Megan, sorry, can we go back? I'm pushing the back button and it's going forward. Sorry about that. That's very kind. Oh, go there. I see. Nope, you're going. Yep, yes, there, right there.
Right here?
Don't stop. I see the arrow now. I'm
Okay.
perfect. I'm glad I have my tech person right here. So this is Tara. This is one of these many hundreds of hydrothermal veins that were subterranean that run as feeders up to the surface in the Dresser Formation. There's at least one person here who I know knows this area very well. Anyway, Tara's PhD was to run around over a huge area and trace these feeder veins and try to go to the surface and look for terrestrial hot springs. You know, how much terrestrial land surface was there back then in the Archean? And she found this type of rock right up here at the top, which Malcolm Walter, a paleobiologist, looked at and said, that looks like geyserite, which can only form in subaerial vent hot spring related environments. And so the middle panel here are all close-ups of Tara's supposed geyserite, and the bottom are geyserites from Iceland and New Zealand, and some data. For example, the black layers that you see there are titanium enriched, for example. And these are associated with stromatolites, closely spatially associated with stromatolites, like you can see there in the upper right. Okay, now that I see where Megan was, let's see if I can actually move forward safely. So this is the current work being done by a grad student of mine on some new core. Is just kind of confirming the basic idea of this painting is from the Smithsonian, and you see hot springs very, very close to stromatolites, which are in either shallow lakes or shallow marine environments. And there's a lot of, supporting other data around trying to understand the environments of early life. So the geyserite, if it is geyserite from the Pilbara, tells us there was emergent land surfaces and potentially terrestrial hot springs. And there is a model out there now that really it suggests that maybe life originated on land for the wetting and drying cycles and then migrated into the sea later. So it's the opposite of what we've all been taught out of the textbooks as a hypothesis. So before I move on then, I just wanted to say a couple of words about stromatolites. And that is that they're mineral microbe layers formed entombing extremophile microbes in the case of hot springs, in the case of hot springs silica. And it's the environment in which they live that they're being entombed. So they're important in that they're not transported and they form something quite distinctive texturally that you can see with the naked eye. You can go in and do a bunch of interrogation on the microscale, but you find them by going, that looks like a stromatolite. And I'm going to come back to that point a little later when we talk about biosignatures. So they're the holy grail in the search for early life on Earth and Mars. No worries. I'm worried my phone is going to do that at any moment. And the question is, do these things show in hot springs preserve into the geological record? And here we see modern Whakarewa Center forming in a relatively deep pool. And this one is a Miocene one from New Zealand that has many amazing features that I won't get into here. But the problem is this quality of preservation is the main issue. Does it preserve well enough through deep time that you can still recognize that it's a stromatolite? In this case, the answer is yes. But when you get into older rocks, can you do it? And 3 weeks ago, I was in Nevada. This is an economic geologist standing on these rocks, telling us that these are reeds, as in plants. And they are absolutely not reeds. The area was about the size of this room. And it's an ancient hot pool full of these conical tufted stromatolites. So it's very, very easy if you don't know what you're looking for to miss the fact that you're right in the middle of a vent, which is very important for them because they're looking for gold. And the gold comes out at the bottom. And if you're in the wrong spot, you won't actually know what you're looking for. You won't be able to find what you're looking for. So this is some new work that we're doing out in Nevada. The other reason it matters is that we spend a lot of money going to places like Mars to look for life. And in the lead up to the 2020 Mars mission, people were using orbital data to construct paleoenvironmental models for the 3 remaining landing sites. This was actually in 2018. They had already then chosen by 2019, they had chosen Jezero Crater, which is of course where the current mission is operating. And there you see a delta, water going into a lake, a reconstruction, and they're suggesting that maybe we'll also find stromatolites in this press release. We may find stromatolites in Paleo Lake Jezero. And then by 2021, as we get closer to arriving on Mars, we see suggestions and comparisons with lake stromatolites from 2.7 billion-year-old Tumbiana in Australia again, and maybe finding these fossils, these living fossils on Mars. And this is true for Jezero as it is true for anywhere. Paleoenvironmental models are only as good as the data you can use to construct them. And for sure, you cannot see a stromatolite from an orbiter in space. So this is an issue for anybody exploring Mars, and it's a continuing issue. I'm not specifically pointing out Jezero. So then if we just really go into this early Earth on the left versus early Mars on the right, we know that, 3.5 billion years ago, both planets didn't have oxygen in the atmosphere, free oxygen. They had their volcanoes and extraterrestrial impacts providing heat. They had liquid running water on the surface. And on Earth, we had what looked to be terrestrial hot springs. We certainly had undersea springs as well. And where there's water, there's life here. So the question, of course, is, would this be the case on Mars as well? And so along comes Spirit rover in the twin rover missions of the early 2000s. And Spirit, many of you will know, had a bum wheel and it was driving backwards, and it basically was dragging its way through the regolith, and suddenly it uncovered a bunch of white stuff. And then they found the actual deposits. Here's Spirit over here in this picture. Let me find this pointer. Let's see if I can find the pointer. Oh, there you are. So here's Spirit, and all these dashed areas are this silica that it found, opal, non-crystalline opal. And you can see that it's actually kind of knobby and bunchy, and in fact, it's finger-like material. It's got a particular texture known as digitate silica. And so they found this distribution of silica alongside home plate itself, which is an ash fall deposit, so volcanic terrain. And the thermal emission spectra, some of you will know this, found a close match to hydrothermal silica that was on Earth, siliceous center deposits, opal plus salt. So the black at the top is the home plate, silica outcrop, and then those are some Earth analogs, and some are washed, some aren't, some have the halite, some don't. And the closest spectral match is from El Tatio, which is in Chile. This is a high altitude geothermal field that's active in the Andes today. And Steve Ruff and Jack Farmer at Arizona State University published a paper in 2016 suggesting that maybe because we know that at El Tatio we have stromatolites, maybe this silica is similar. It's the most Mars-like of the Earth analog settings. It's at high elevation, low water flow, cold, arid, thin atmosphere. And so they found indeed microbes in layers in those knobby things. That's debated by some, and I won't go into the details too much. But the other thing they suggested was that just to the west, circled there in red, is Pioneer Mound, which they now believe may possibly be a remnant paleo vent, shown on the left up there. Now that's being compared to Chilean hot springs. The rover died, and they never were able to drive over there. It got stuck over on the other side of where you see it in the picture. But we can use environmental models to at least consider what we should find if anybody drove over to that possible vent deposit. We can use the models that I've been talking to you about. And it would help because, it's a lot of money to go to Mars. So we decided in New Zealand, since it was much more accessible to get to hot springs in New Zealand, you even have an Airbnb nearby, and you're not at high elevation. So we started working on digitate or hot springs in New Zealand. There on the left is a New Zealand example, and on the right is some of this Martian silica, which, by the way, is a hydrothermal silica, but we have not confirmed that it's sinter yet because we don't have any samples. So if you look at this model, you can see I've boxed up where you find the digitate sinter, and it always forms in a certain environment. It can be knobby or it can be, narrow and spicular, but it's digitate. And you find them always at subaerial growth interfaces, always at the air-water interface, always growing subaerially. So there might be water right there, and the water is weakening up by cooling and evaporation, bringing the silica up, and the microbes are layering and layering up, and you build up a stromatolite. So that's where digitate sinter forms. But we went further, and I had a student who, Kim Hambley, who worked on growing these things from scratch at Champagne Pool, which is the most famous hot spring in New Zealand, just like the one you saw in Yellowstone, except this one is called Champagne Pool because of all the carbon dioxide bubbles that are constantly bubbling off this pool. And the center is the white stuff growing around the rim. So Kim put out glass slides and grew sinter hourly, daily, weekly, monthly, and multi-monthly during her master's. And we had a weather station set up nearby to look at the wave and wind direction because the wave action in the pool, she wanted to know, do the sinters grow faster over here or slower over there? And looking at wetting and drying cycles and evaporation, and grew this layers of stromatolites. So here's the most important thing for me, and it's really, in my view, super critical. And that is, this is one of the slides she grew. And you can see that there are layers of filaments, FIL, and then solid layers of silica that are the black arrows. And there are also little pore spaces that are black, known as fenestre, which means window in Italian, so open. And so those might be microbially mediated. But we assumed that you had alternating organic and inorganic growth on those digitate sinter. So we thought the filaments were the organic phase, and then maybe seasonally you get the solid silica or sinter lamina, which is very thin layer, and then another season. So that's what we thought, and we were super wrong. But that was our idea. What Kim did next was amazing. Kim was working with the microbiologists, and they did experiments. They used fluorescent molecular probes, and they looked at disruption techniques to determine, in fact, and imaging, determine that the solid layers that you saw that we thought were inorganic were actually EPS, exopolymeric substance. Actually, the goo, the snot that microbes will give off to sort of stabilize their environment and to make it a better place to live. So those layers of EPS silicify and they make lamination. And why does that matter? Who cares? Well, it matters because the whole thing is biology. All of it. All of the digitate stuff growing here is biology. Even the stuff we geologists thought wasn't biology. All of it is entombed in silica. It's an astounding result and has really been an amazing finding in my view. So then we went on with our wonderful student from Thailand, Kitty Sriaporn, who did some amazing work looking at a lot of digitate sinter in New Zealand at all different fluid chemistries. Acid sulfate all the way down to alkali chloride. And there they are, all growing at the air-water interface, found in all fluid spring types and actually at all temperatures, always in the same general environmental setting. Okay. Now I'm going to do a tiny segue and just tell you why are all those fluids different? Tiny little segue here. So we've got a magma chamber down below, the surface up above, groundwater circulating, hits the heat of the magma chamber, and of course rises by convective heating with water rock interactions, picking up dissolved silica, chloride, etc., from the magmatic degassing and the country rock. And then you get to a point where it's not so so much overburden. It's not as heavy. And so the water starts to boil, which is those black circles you see near the surface. And if the water goes straight up to the surface, then you get a hot spring with geysers and very, very thick silica, very thick sinter. And that's this over on the right, the blue. So that's these chloride water springs full of cyanobacteria, mainly what I've been showing you. But if the water boils and the water then that flows sort of out instead of up, then you have acidic steam separation. carrying hydrogen sulfide and making the red area that you see that's full of sulfides and stinky mud pools, fumaroles, mud pots, and acid springs. That is with very thin centers. This is what NASA was freaking out about, I'll have to say, at the landing site meetings in 2017 and 2018. They were really worried that you'd end up with something on Mars that was more like the red and not like the blue shown here, where you wouldn't have abundant life. We might not be able to tell. It might just be a yucky, acid, terrible place. And so that's why Kitty was looking at all the springs to make sure, well, is there life in all these digitate things or not? And so, of course, what she found was there was life in every type of spring at every temperature. So there you see the digitate features on the left with the different spring water types. In the middle, you see the laminations shown by the red arrows. Laminations you need to prove that you have a stromatolite. They're laminated. And then on the right are all the different microbial communities. So we have universal layering and different microbial communities controlled by pH and temperature. So we have Pac-Man, and then we have rods, and we have filaments, and we have a lot of stuff. And not all of it, but at least some of it gets silicified. So yes, it's true, those are different organism communities, but they all make stromatolites. So Kitty showed really nicely that it doesn't matter if it's an acid spring or a mix or whatever, it's still a stromatolite. And the one on the left is what the product is when you're in an acid spring, which is quite nasty. This is at Hell's Gate, sulfur, etc., stinky, muddy. And then over here we have our beautiful Arachicaraca with all those gorgeous mats. That's an alkali chloride spring. It's constructional instead of destructional. It's got a more neutral pH. And thank goodness that hot springs algae sign is now being buried by center because they're not algae, they're cyanobacteria. And obviously that was put up there like in the 80s. And it's now, since the time I've been in New Zealand, it's been completely buried, thank goodness. Okay, so NASA was very worried about us finding this on Mars instead of that. But it turns out that we have life everywhere in hot springs. So let's go to Mars. Here's Husband Hill, part of the Columbia Hills. Home plate is down below. You can see it here. The rover rovered around and found a bunch of sulfur-rich soils, which is what they were saying. Oh my gosh, it's an acid system. But remember, the rover also found silica. And so the question was, is that okay? Is that a problem? And we got sulfur-rich soil and we got opal-A silica, which can be acid or alkali. Can they both form from hot spring activity? The answer is absolutely they can, because when the water table goes up and down, we can switch from acid, steam to alkali chloride or a mixture, and it's still okay. It's still a hot spring system and you could still preserve life within it. But this was an issue and definitely an issue in 2017 and 2018. They were very worried about it. So Kitty went on, and I'm not going to explain all the microbiology. Remember, I'm a geologist. But let me show you, and I just want you to look at all these beautiful colors. This was our microbiology student. So here we see along the bottom, we see those are different locations in New Zealand. And the more alkali chloride springs are to the right, and the more acid springs are to the left. And she found lots and lots of microbial communities, but they're different when you change chemistry, as I told you before. And just one example here is in the alkali chloride spring way over on the right. This bright yellow are cyanobacteria. And you don't really see those in the acid springs. You get more diatoms and other things. Acidophilic algae, for example, in the Pac-Man is actually an alga. So we won't go into all these details. She did a lot of really interesting work. And also found that there were adaptations to high temperatures like genome streamlining, for those of you who are biologists, and thermostable amino acids and nitrogenous bases. So we're pretty excited about this because, again, life is pretty much everywhere in digitate center. So that brings me to the 2020 mission. The mission goal and aim was to explore a site likely to have been habitable and seek evidence of past life. And of course, they chose the poster child over there, the Delta. But they also we were looking at impact basins as well as Columbia Hills. I was very honored actually that our site got all the way to the final 3. But my take on all of this, being an outsider, is that it's pretty hard to explain to people why you would want to go back anywhere on Mars, like us geologists who go back all the time to learn new things. It's very expensive, and I can imagine that going back is really not that exciting when you aren't a geologist, I think. I don't know, but for sure that delta was calling. And so that's where, as you know, the current mission resides. So NASA went to Jezero and we went to Japan. We took the Mars LifeSpring mission concept to Japan and had a workshop in December 2019. Here's a JAXA engineer presenting a feasibility study of actually going and getting those samples on Mars outside of a standard NASA mission. JAXA, you may recall, is really darn good at getting samples back to Earth from asteroids. And the Hayabusa 2 mission brought back carbon-rich samples from Ryugu, an asteroid that turns out to be a dead comet, and it has 5% organic matter in it. It's black. It's full of organic carbon. So anyway, they have all this legacy, and we were meeting in December of 2019. This is the engineers talking a lot about how we would sample. I don't care. I'm a geologist. I just want to take a rock hammer up there, smash it, take a sample and bring it back. But they have all these ideas. But just to show you the size of the sample that we're after is a 9-gram sample that is intact texturally, because we need the spatial relationships to look at, well, is it like what we have on Earth or not? So we were discussing this at length in December 2019. And then this slide was put up looking and comparing a NASA mission and costs of the NASA mission, which I've updated based on recent estimates, which that mission for sample return is now on hold from Jezero because of that price tag and how long it's going to take to get the samples back. And then he was comparing this to a Japan, ESA, Australia, New Zealand, US mission, looking to return to a known site, focus on a grab and go mission, make it nimble, make it cheaper, and make it science driven. But I kind of also wanted to point out something else about this slide. And that is this part right here. I was sitting in this workshop and I thought, what is that? You know, what is that samurai spirit? I didn't really know. And I was like, okay, is he kidding or is this what is that? So I looked it up and I find that those with a samurai spirit are capable of serenely carrying out their work in the face of adversity and have the willpower to master themselves. And when I realized what he was really talking about, which was a Japanese cultural view of a space mission, I was like, I'm in, I'm doing this. This is what I want to do. I want to be involved with a mission that isn't a standard NASA mission. This is something else. And it's imbued in all the way that we've interacted with these scientists and engineers. But as you can see, that was December 2019. And you can imagine what happened next. So 3 years later, after New Zealand was in lockdown for almost 3 years, isolated from the rest of the world, we all got together again at my house. That's my house, that's the backyard. And we finally got the group back together again for a workshop. We had to eat first, of course, so there's a picture of the food. But we had to do a workshop and then we went out in the field. And the engineers have been sitting around in their houses for like 2 years and thinking about how to sample, because some of you know about the InSight mission where they were unable to stick a heat probe down because of the hard pan that's on the ground there at the InSight site. And so we don't want that to happen. We don't want to go to Mars and then find out, gosh, we can't sample. And some digitate center like this is hard as the hobs of hell, as they say in New Zealand. Some of you will know what I'm talking about. This is so hard that you have to use a sledgehammer to get that stuff out. Other digitate center is so easy to pick up. You could pick it up with like a postage, like a Post-it, you could pick it up. So we have a lot of concern about that. And we're trying to get a study funded to actually work on the aspects of the sites in terms of sort of what are these materials made of? So we had a student work on some of this digitate center, and this is from El Tatio. This is a micro CT scan, looking at porosity and trying to relate that to the microbes and the other features of these digitate centers to try to understand better about how you might sample them. Because the last thing we want is to get all the way to Mars and not be able to actually pick up those samples. So anyway, Jody will know kind of what I'm talking about. But so this is some work that was published in 2023 by one of our honors students. She did this work as an honors student. So the very, very last thing And I appreciate that a few of you might be dashing off, but we've got a few minutes left, and I really want to talk about this last issue. And there are some people in this audience who know exactly what this problem is, and that is what actually constitutes a genuine, bona fide biosignature. We're all talking about finding life on Mars, fossil life. What's it going to look like? And in 2018, this paper was published, and they made this thing called the ladder of life detection. So I've color coded it for you. The biologists, this is from a very biological point of view, by the way, and they stuck all the life stuff up at the top. So living things. What is life? Life can be Darwinian evolution evidence. It can be growth and reproduction. It can be metabolism and evidence for metabolism. And then they stuck the geological stuff down at the bottom and called us suspicious. That hurt my feelings a lot. I was like, suspicious biomaterials. That doesn't sound nice, you know? It doesn't. And that's all the purple stuff, okay? And look what's at the very bottom. The thing that I work on right here, bottom of the ladder. You can see why it hurt my feelings a little bit, right? Well, okay, I know, but I'm going to acknowledge some stuff in a minute. But these are considered suspicious and they're at the bottom. And that includes all kinds of other things that I'll get to in a minute, because I'm going to spend the very last bit just talking about what do we do about this ladder of life detection. There are some assumptions being made here by the biologists that I will also talk about. So some of us believe actually, well, I'll give you the punchline in a minute. Let me just show you some stuff. So the truth is, and here's where I'm going to admit to the problem. The problem is the quality of the preservation of the biosignature. Number one, you get into the deep time geological record, you go back really far and you start to lose your sense of what paleoenvironments are you in, for example. So the quality is the problem. And if we take that lovely mat and we go to a 6,000-year-old mat on the right, and then we drop down, that's another bit of the 6,000-year-old mat. And then you see, you start to get patchy preservation of that palisade fabric. That's a problem. And in fact, this palisade fabric here, some of it's been turned to quartz from opal. And you would have trouble, even I would have trouble realizing that that lower left white stuff was actually a microbial mat. Okay, it's true. I admit it, and it's only 6,000 years old. So the issue is this diagenesis. That's part of the problem. And diagenesis is all the physical and chemical changes that turn sediment, for example, into rock. And it goes through compaction, cementation, recrystallization, and replacement, and you could end up with this. So that's a little scary. And so we go through opal, which is non-crystalline. This is XRD data for mineralogy. And then you go through paracrystalline phases, and then finally microcrystalline quartz, which can destroy your textural biosignature. It is true. I have to admit to that. And these are just some pictures just so you can really see, because of course we've worked on this a lot. So I might as well show you. On the lower left is an absolutely beautiful cryo-SEM image of a living mat that has both the filaments, the little balls of opal that are just starting to form. It's really baby rock. And then the sort of shaggy stuff is the EPS. So it's all preserved. But then you start marching through diagenesis and you end up, this is only half of it, end up with this cristobalite tritomite, and you begin to lose the integrity of your mat. And it can get even worse, but I won't show you because it makes me have nightmares at night, so I don't want to show you. It'll upset me. So the other issue besides diagenesis is the environment that the rock sits in for a certain amount of time. And this is a classic example in New Zealand at Tekopia. We have a 400-meter offset of a fault that has acid steam alteration all over the place related to the fault. The water table dropped, but before that, we had hot springs that were forming beautiful alkali chloride sinters, geyserite, microbial fabric, and they're all now sitting today, 5,000 years later, in a steaming landslide, being eaten alive from the outside in. They're being destroyed by the acid. So this system didn't shut down fast enough, and it's sitting around cooking the hot spring microbes, and they are corroding, and they're going to disappear. They're just going to be eaten away. So the same process that formed the hot springs is now destroying the center. So you must understand your environmental context if you really want to find good preservation. Well, that's depressing. That's depressing. However, all is not lost. We can go to much older center in the Jurassic of Argentina where I work, and you can find exceptional preservation of center. Here's Quartzo center, the green being the Carrageen carbon, beautifully preserved, well, for Jurassic. And that's cool. And you can even find in the same place, insect eyeballs perfectly preserved, bristles of little insect arms, I don't know, legs with bristles perfectly preserved. Really fantastic. How does this work? Well, this is this ongoing question that I'm really interested in. And so what we're talking about are Lagerstätte, exceptional mother lode style preservation of fossils. We want to find those things on Mars. We want to find those things in early Earth. We want to find the best preservation, and it's patchy as hell. And how do you find it? Well, you got to go to the outcrops and you got to go to the geology and you got to figure out, well, where are the ideal conditions regionally and then locally, and then at the micron scale, where do you find the best stuff? And this is Claudia. This is a hot spring deposit in the Jurassic of Argentina at Loma Alta. So the number one solution to sorting out this problem of preservation is that you must have well-constrained paleoenvironmental context, first and foremost, and it has to be at the large scale first. And so if you look at an actual geological map, we're down in Santa Cruz province at the bottom of Patagonia, and we see there are these big block faults. So the stuff that's pink on the top is Cerro Vanguardia. It is the biggest gold mine in this region. It cranks out loads of gold and silver. It's the plumbing of a hot spring system that has been faulted up, and you find all the gold in those veins that are red. Block faulting down, and you find the Claudia deposit. It's to the south east, if I know my north-south, and this is Loma Alta. And notice that it's at the intersection of two faults, and the only reason you can see it is that a river has eroded through it, the Seco River. And there it is. It's a big hot spring system. So you would not have this much gold if you didn't have a lot of fluid and a lot of water, either a lot of it in a short amount of time or over a long period of time. And so this may be a clue to why we get such great preservation at the place where the two faults intersect, where fluids may have been coming up in that Claudia deposit to the southeast. So I don't know if that's true, but we have exceptional preservation at Loma Alta. And that's the geology. So that's the first thing we do is go, what is our context here? Where do we find the best stuff? And then key number two, you must have multiple lines of evidence to prove a biosignature. You can't just say it looks like a stromatolite. And so this is beautiful work done by Bonnie Teese, who's now a postdoc at JPL, who's an organic geochemist from the University of New South Wales. Exceptional work that she's done. And at Claudia, she found phenomenal preservation of different kinds of organic molecules, in particular hopanes and steranes. And suddenly, look, we get to go up the rung of this ladder of life detection. We're right up here at this part, organics not found abiotically. Right there, we're a little bit closer to being able to prove that this might be life. And we know this is a hot spring, right? We already know this. So it's when you're not sure, that's where the issue is. You can step up a little bit more by finding pigments, which my master's student, Emma, did. And that's at El Tatio. The red being some Raman work showing some certain pigments. They're shown in blue and orange there that compare to some salars, some salty areas in Chile that are other extreme environments. And so we have these silicified filaments that have their pigments still preserved. So we get to go a little higher on the rung of the ladder of life detection. And we actually mapped this stuff out with a synchrotron and Raman, and found that these filaments have areas of high and low intensity of these materials, which might be suggesting early diagenesis already when they're in hardly rock. So we already are seeing the beginning maybe of diagenesis because we don't have even preservation of those pigments. And the final piece of work I want to point out, and then I'll just wrap up, is this nice new work we're doing with the synchrotron micro XRF over in Australia and Melbourne. And this is my student Barb who's doing this work, and you can see Palisade Center. It's about 15,000 years old in New Zealand. looking at another type of biosignature, a multiple line of evidence, and that's trace metal distributions. You can see iron shown there in red and gallium shown in green. And the gallium clearly outlines the sheaths of these cyanobacteria fossils in a Palisade microbial mat. That's about 15,000 years ago. So if you lose your organics, would you preserve a trace metal signature of some kind? And the answer is, well, maybe it's in gallium. We know it goes back at least to the Jurassic based on pilot studies. And so, oh, look, we got knocked down the ladder again. Gosh. Here I am down here at potential metabolic byproducts, you know, nickel, cobalt. We could add gallium here as well. We have a Marsden or NSF style grant in right now, waiting to hear whether we're funded to look at gallium isotopes and gallium concentrations in centers in Iceland, New Zealand, and Yellowstone. So we'll see what happens. We're hoping this is actually a biosignature that's outlining these sheaths that are made up basically of polysaccharides and lipids. So to bring this all together, the ladder of life detection to me is kind of bunk. And I mean that not in a mean way, but actually as a geologist, I would flip that ladder of life detection and make it a pyramid of life detection with textural biosignatures right up at the top. The first thing that you should look for is something to narrow down where you should look. And what is that but a texture? 100%. We go in the field and go, my gosh, that looks like a stromatolite. And yes, you need to test whether you have a biomorph, which is a pseudo fossil. Is it fake? Is it not really? It just looks like a stromatolite. You must do what I'm talking about with these multiple lines of evidence in order to constrain whether or not you have a bona fide biosignature in the geological record in these hot spring environments. You're never going to have Darwinian evolution evidence or growth and reproduction evidence in a fossil in the geological record. So this does not work for us geologists at all. And what we need to do is say, look, I respect that totally, but we need to start with the textures and then test, and be sure that what you think you have, you have. All right, so in summary then, I'm just wrapping up right now. I think you might see that I'm really interested in using paleoenvironmental context and models to help us, plus the preservation state, which you must check for, to help us narrow that search for biosignatures on early Earth or Martian rocks. I think that the digitate silica on Mars is a really good place to go, and the analog environment are the environments I've been showing you, and that we need sample return to apply these multiple techniques, which a rover can never do. So maybe on that note, I will finish and say thank you for your attention, and I'm happy to take questions. Thank you very much. Megan, thanks for saving me earlier. Yeah. So between Jody and me, maybe we can take some questions.
I'm just
Go for it. Are you helping?
I'm just a wrangler.
Thank you, Wrangler. Any
Megan
questions?
over there.
Megan, please.
Actually, a question. So I guess I have two questions. One is when it comes to the River Delta and Jezreel Crater versus Columbia Hills, I'm a little bit curious as to like what the benefits were being weighed out in terms of the science, not necessarily the politics. And
Right.
my other question is when it comes to sample return from Mars, which is obviously faces a lot of challenges, how well are you able to search for multiple lines of evidence and what you expect to get?
All right, now help me remember the second one. The first one for people online is what was the sort of Jezreel versus Columbia Hills? I think there were a lot of open questions in 2018 about Columbia Hills, about home plate that we've been working on since 2018 to try to rectify the concerns. And one that is still wildly open and widely open is, can you form digitate sinter without any life at all? And it is impossible to do that in a natural hot spring. There is life everywhere. I won't say which one of my students wanted to dump like some kind of poison, some kind of antibacterial in a hot spring, but we are not allowed to do that in New Zealand. And I doubt very much that you'd be allowed to do that. That requires then experiments in a lab. And I'm aware of, I think, 2 labs that are working on this problem of, can you grow this stuff inorganically? Which will still not negate the possibility that there might be life preserved in the silica on Mars, but it means that it could be one or the other. In regarding Jezero, I am not, privy to all the work that's being done, but I do know that at the time we suggested that the delta was quite young, and they were very hooked on the idea that the delta was quite old. Turns out the delta is quite young, which is why it looks so pretty. It's not, we wrote it away. And so it's in the Hesperian, not the Noachian, which means it's in the sort of outburst flood stage, less water kind of stage. And that is not widely discussed, but it is actually now been shown separately to be younger. And it's hard to date things on Mars's surface. I understand that. The other thing they were hoping to find was organics out in mud or clay out at the edge of that delta. And they found no mud, no clay. And that's because it's a Gilbert delta, not a Mississippi style delta. And a Gilbert delta are deltas that are very coarse, form very quickly, almost like outburst floods. So less, how do I say this? An environment that might be less conducive to life establishing and taking hold. So as far as I understand, they are now up on the rim of the crater itself, hoping to find carbonates and stromatolites. And I very much hope they do, I do. But my feeling, as in 2018 and 2017, as it is today, is that I think this is an exceptional place to look for life. If you don't find life at Columbia Hills home plate, then I really worry about finding life anywhere on Mars, because it really should be at the Columbia Hills digitate silica. So my take on it is, I hope they find signs of life. We want to do that. The mission's supposed to do that. But the delta, there's some issues with the delta. So the focus is now on the carbonates as far as I understand. So that's how it's evolved from my outside point of view, looking in. And now the second question was?
Second question was, how well are you gonna be able to search for multiple lines of evidence with the amount of material you'll be able to return?
Right, so the rovers themselves are not gonna be able to do all this stuff, although of course they have nice, really, they can do a lot of nice things. The samples need to be, they've calculated, our engineers and the scientists, that we need at least about 9 grams per sample. And geologists always look to the biologists for how many samples you need. And everything I've ever been told is at least 3. You got to have replication. So, that's what probably would happen if that mission was ever to come to light, which would have to be supported commercially. ESA is interested, but they're busy with the Rosalind Franklin mission and the ExoMars. So, they're interested as well. Australia and New Zealand don't have any money, so we can't just launch a giant mission. NASA's very busy dealing with its own issues. So we don't know if this mission will happen. But what I do know is that it'll be a lot cheaper, because we already have been there and we know the landscape. And so, no, we wouldn't go back with a rover with lots of instruments. The whole idea was to get those samples and come back. And the final point that at least one other grad student made to me today, which I agree with, is that the minute China decides to get a sample from Mars, suddenly we'll have money to go to Mars and get a sample. And I really hope we go to Columbia Hills.
Other questions?
Do you know what the silica is doing, like, in the biology?
Do I know what the silica is doing in the biology? What a great question. So there's been lots of discussion about whether that silica is just passively templating on those microbes, or whether there's some sort of active thing going on. And from my point of view, it doesn't matter, because I'm interested in the preservation. And I like it that it goes straight on there. And there are other elements besides gallium that are also being attracted. I didn't go into all that because it gets a bit tedious, but there are a lot of things we can test out and look for. So whether it's passive templating or active, from my point of view, doesn't matter. We think it's mainly passive. It's passively getting stuck in the silica. And microbes do have ways of sort of migrating up through and starting over and making layers. So whether it's active or passive isn't such a concern of mine, but it is a concern of some. And sure, it'd be nice to know. But, and the metals, it's the same thing. What is actually attracting the metals? Is it really the organics, or is there something else that's actually attracting why the metals are there? Are they needed metabolically, or is it just they glom onto the EPS because it's a great attractant? And there's been some work done on that. But again, from my point of view, if we get a biosignature that looks robust, well, let's go for it. That's my view on that.
Any questions?
How similar are like silica versus carbonate textures of those?
How similar are the silica versus carbonate textures? Super great question. Remember, there are travertines also. And our experience is a lot of travertines where we work in South America, and they do not preserve well unless there is early silicification. And most early life stromatolites are, a lot of them were carbonate, but they've been silicified early. So one of the next steps in our research is to understand, well, what do you mean by early? And, what else do you need to get exceptional preservation? The textures in the carbonates, generally speaking, are not as good unless they've been silicified early. Like, riding shirt type stuff. Yeah, sorry.
How well is, because the pictures you showed of the digitated silica on Mars weren't like by an active hot spring. Are all the ones we find on Earth that have this like morphology of the digitate by active hot springs, or can they preserve really well like later?
The question is when do these digitate things preserve really well, especially in the modern hot springs where we know what's going on? And it turns out that we don't see digitate center very much in the geological record. So a lot of our work into the record, as I mentioned, is on the Palisade fabric, which preserves quite well. The digitate, as you can see, is sticking out. It's subaerial. It's not in the best sub-environment to preserve. I've seen some in the Cretaceous centers in China, and a few other places, but it's like, let me put it this way. The vent area, it's like a point, okay? The vent area is a point, and so subaerial vents don't preserve very well. They are subaerial. They get eroded and weathered and stuff happens to them. Steam comes up the vent when they die, and then you get all acidified. So the vent is a small area and it doesn't preserve well, but what preserves well are the aprons and the marshes. So we have this sort of volumetric situation where we don't actually even preserve the vents. And so it's sort of the same with the digitate stuff. It doesn't preserve into the record that well. But what we do have is all the other textures preserved beautifully. And so, yes, we don't always see the digitate, but we definitely see all the other textures. And so I think we do a lot of our work on the digitates, but also on those textures that will preserve into deep time. Yeah. Yes.
If you manage to go to Mars and grab the digitates.
Yes.
Bring them back somehow without them actually being destroyed on the way back, actually.
Yes.
What would you then do to like check it to see if it has like?
Yeah. So the question was, you grab this stuff on Mars and you bring it back. Hopefully it's not broken. So one of the things the engineers suggested is that we get regolith and we put this thing, I don't know, in a box, and then we dump a ton of regolith on it and just stick it in a bunch of dirt and bring it back, Martian dirt, because you don't want to break it on the way home. We want to keep them intact. No drilling, powdering, that kind of thing. And then the kinds of analyses that I mentioned to you today, but also the Japanese have this astounding laboratory in the middle of nowhere in Japan. And they have more equipment than any of you could imagine all in one place. And they've done an incredible job on the Ryugu asteroid comet. And so they have super high-end, exotic isotope labs and the whole thing. So I am confident that we would develop more techniques with them and others involved once these samples are back to Earth. So you have to, as you see, you have to have multiple lines of evidence, not just, it looks like a center. It looks like a stromatolite.
Really?
Finished?
Right, oh, another one.
One more.
That's like
Yes,
so on Mars, there are these digitized samples.
yes,
You are claiming that may only be produced by biology. And so
perhaps, yes.
you-
I won't
okay.
commit. Yeah, so
Just want to make sure I got that.
yes, so what has to happen, and it won't be by me personally, is that we need to grow these things in the lab and find out whether you can grow digitate silica in a hot spring environment without life. And you cannot do that in a modern hot spring at all. There's life everywhere. It's impossible. Life is everywhere. So the answer is, we don't know if you can grow them abiotically. Maybe the stuff on Mars grew abiotically, but what a great place to go and check it out, because on Earth, they all grow biologically influenced. Yeah. Great.
Okay, thank you,
Thank you.
Kathy. And Kathy's around for a couple more days. You can contact me if you want to See her, and she's giving the
Paleo lunch,
paleo lunch at the Burke Museum on Thursday. It'll be completely different.
totally different
on a
topic.
Totally different topic, but paleoecology.
Yes, from New Zealand.
Yeah, also from New Zealand.
Yep.
And so thank you very much, Kathy.
Thank you, Jody. Thank you, everyone.